children, in the previous section we have done that how from the anther primordium the MMC have developed, that is the microspore mother cell, and from the microspore mother cells how the microspores developed. The MMC, that is the microspore mother cell, which is diploid structure, when forms microspore by undergoing some mitosis and meiosis, when it forms the microspores, this process is called as microsporogenesis. The microsporogenesis, along with the diagrams, all the steps, is asked in exams many times under a 3-mark question and all. Now, in the next section, what we have to do is, from the microspore, how a pollen grain is formed. That means, now in this section, we will discuss that how the maturation of the microspore occurs and it changes into a pollen grain. We have got the microspore structure after meiosis. That means the callose dissolution takes place, and from a tetrad, 4 microspores we get. Microspore is haploid because it has undergone, it is a resultant of a meiosis. Microspore is having a big nucleus and a dense cytoplasm. In the very next step, it undergoes vacuolization. Vacuolization means formation of vacuoles. The big vacuoles occurs, and these vacuoles then get filled up with the storage, what you call, substances. In this, when vacuolization occurs, this structure increases in the size. Immediately when it increases in the size, it undergoes a asymmetric division, again which is a type of a mitotic division. Asymmetric division means one cell will be larger than the other. The larger cell is called as a tube cell or a vegetative cell, and the nucleus is called as a tube nucleus or a vegetative nucleus. These tube cells have vacuoles, and the lower cell, which is the smaller one, is termed as generative cell, and the nucleus is called as a generative nucleus. This generative cell do not have any vacuoles. Only the upper, the larger tube cell has the vacuoles. After this, what happens is, there is a formation of a pollen grain, and first of all, the pollen grain, what you call dermis, forms. This dermis, this outer layer, is made up of exine, outer part, and intine. In is inside, ex is outside. Exine and intine are the layer of a pollen grain, and together it is called as sporoderm. The term is given as sporoderm. Exine is made up of sporopollenin, which is the most resistant substance in the world, means no chemical can degrade it, and we all know that sporopollenin was made up from the Ubish granules, which was made by tapetum layer. This exine, sporopollenin, and intine, very importantly, intine is made up of 2 things. One is cellulose, another is pectin. Remember this NCERT line. This topic is given on page number 23 NCERT. Just go through it. Now, intine, inner layer, exine, outer. I told you exine is made up of sporopollenin, the most resistant substance, and intine is made up of cellulose and pectin. It's the same thing, the tube cell, tube nucleus, 2 vacuoles filled with the storage tissues such as starch, proteins and lipids. We have the generative nucleus and a generative cell. At this point, the this pollen grain, which is of course the end structure. It has developed from the simple mitosis from the microspore. It will be haploid therefore. This pollen grain structure is called as two-cell stage. You must have seen here, we have two cells here in the pollen grain. This is called as two-celled structure. Most of the angiosperms, that is 60% around, in this case, the pollen grain is shed from the anther at this structure, this point, that is the two-cell structure. In the rest 30%, remaining 30%, this pollen grain is shed at three-celled structure. Three-cell structure means that the generative cell, which is the lower one, the smaller one, the generative nucleus is formed into two male gametes, which will fertilize the ovule. Therefore, one tube nucleus is here. The generative cell is what you call divided into two nucleus, more two nucleus. These are called as male gametes, and that is why these are called as, this is called as three-celled structure. Exine at the outer one. At this point, it is shed in very few, that is the 30% of the angiosperms. This is called as three-celled structure. This is called as two-cell structure. In the previous section, I have told you that from the microspore, when pollen grain forms, this is a precocious development. Precocious development means the microspore starts germinating inside the anther only. And at this point, when it germinates, it forms the two-cell structures and it is what you call shed at this point. Sometimes, only in the few cases of angiosperms, it is shed at three-celled structure. That means the generative cell has been given to the two male gametes. Now coming to the exine part again, we know that exine is made up of sporopollen, most resistant what you call substance. Then how the pollen tube emerges from there? For that, exine is basically a porous structure. Exine has a pore or an aperture which is called as a germ pore. This is the germ pore or what you call aperture from where the pollen tube arises. In many cases, like dicots, there are three germ pores. That means one here, second and third. From any of the one germ pore, the pollen tube can arise. Dicot feature, tricolpate. Tricolpate means having three germ pores. That means three pores in the exine layer. This germ pore is not having a sporopollen. If only one germ pore is there, it is a feature of a monocot and it is called as monocolpate. Is that very clear to you? This is called as monocolpate. So these things you have to remember, tricolpate and monocolpate. This pollen grain, when develops into this structure, soon it goes on stigma and a male gametophyte is formed. In the next section, I will discuss how the male gametophyte is formed. Thank you.